Yeah, so hi, I'm Alexandria. I was working in Miguel Pinero's lab this summer. And what I did was a structural functional study of a potassium transporter found in rice. So this protein is a member of the HKT family. It helps mediate the transport of potassium and sodium ions across plant cell membranes, which is involved in plant responses to salinity. It exists as a dimer, which is an interaction between two protein subunits, and this is predicted to occur through salt bridges and hydrogen bonds. And so what I was focused on testing was when you disrupt the salt bridges and you disrupt the hydrogen bonds, do you disrupt the interaction of the transporter, and do you also disrupt its function? So we use the plant system to look at interactions using a technique called IFC. So essentially, we have our two subunits of the HKT with a half of a fluorescent protein appended. And if the subunits of the HKT interact, then the fluorescent protein will come together and fluoresce. So if we see fluorescence, there's interaction. So with the wild type, which isn't, there's no mutations, this is just the regular version, we saw the fluorescence, there's interaction. And when we disrupted the hydrogen bonds, we also did see fluorescence, which indicates disrupting the hydrogen bonds wasn't sufficient to disrupt the interaction. However, when we disrupted the salt bridges, we saw that there was no fluorescence, whether it was tested against itself or the wild type. And we also saw no fluorescence in the double mutant, which indicates that disrupting the salt bridges led to a disruption of the structure of the protein. We also wanted to look at the function of the transporter, and for that, we use a frog egg or oocyte system. So this is some microscopy we did in the frog eggs of the wild type. This is the wild type without any fluorescent tags. This is the wild type with the half fluorescent tags. And this is the wild type with a full fluorescent tag. And what this tells us is that we still see the same interaction in the frog egg as we did in the plant, and that it's localized to the cell membrane, so we can continue on with the system. And then we use 2 electrode voltage clamp, and that is just a way of measuring the transport rates going on inside that frog egg. And what we found is there was no significant differences between the different wild types with the fluorescent proteins appended or not. So the fluorescent protein isn't interfering with the protein's function. And so what we want to do in the future is repeat this functional assay with the other mutants. We're especially interested to see if what happens with the disrupted salt bridges, because it disrupted the structure. Does that also disrupt the function? And overall, this research is important because of the role HKTs may play in plant responses to salinity. And this fundamental knowledge may be a stepping stone in eventually getting more salt-tolerant crops.
Wow, that's awesome research. So fascinating. Thank you for sharing with us. I have a couple of questions for you. So do you think that this phenomena that you see at the bottom here, do you think that that's could be similar across other HKTs? Do you think that that's possible?
Yeah, that's definitely possible because, as a family of proteins, they do share some structural homology. And that's part of why I think this research is important, because other HKTs may have similar interactions with salt bridges and hydrogen bonds if they also exist as dimers. And then that would mean the findings I have here may translate over to the other HKTs in other plant species.
Awesome. And it sounds like you've been busy doing a lot of different experiments this summer. Was there one that you found that was your favorite? This frog egg assay seems so cool.
The frog egg assay is pretty cool, but I think my favorite was definitely the fluorescence microscopy with the tobacco plants, because essentially we had transiently expressed our protein in them by injecting them with bacteria, and then they had to incubate for 3 days, and then we got to take them to the microscope, and just seeing the fluorescence for the first time was it was beautiful, because it's that's all my hard work, and then it came out in this gorgeous image of the plant cells. So I think that was probably my favorite part.
That's awesome. Yes, I will say they're beautiful images. As someone who doesn't do a lot of microscopy, they all look gorgeous. Well, thank you so much for sharing all your hard work this summer. We